Welcome, dear colleagues, distinguished participants, esteemed students, and honored guests. It's our great pleasure to welcome you to the second day of the Virtual International Congress on Computational Biology, Bioinformatics, and Artificial Intelligence. We are going to start. At the program, at the first, in the second day, we have, just a minute. We have here our guest speaker, Dr. Sayed Arshi Yuz Zaman. He comes from University Banashtari, Vidyapit, India. You are here.
Yeah, I'm here.
You're welcome. You can share your presentation. You have the floor. You can start your presentation.
But I cannot see the sharing option here. Can you please tell me
Okay.
to share?
You have the button, green button. Just a minute.
Yeah.
Yeah, you are not here. Just a minute. Just a minute.
Oh, okay.
I want to resolve problem. Now you can do it. You have the floor. You can start your presentation now. You can start here. It's okay for you?
Can I start now?
Yeah, you can.
Oh, okay. Thank you very much. So good morning, everyone. My name is Sayed Arshi, and it's a pleasure to be part of this virtual international congress. And I thank the organization for inviting me to speak about my work. So today I'm going to present you about one of the novel strain of Rhodococcus indonesiensis, that is a SRC1, and it is being used for the petroleum bioremediation. That means that it can degrade the petroleum products and petroleum as such. So now why we are working on this. So the thing is that what are the problems? Like, why a petroleum hydrocarbon is dangerous for us. So most of the time, I mean, we are using a lot of petroleum products, and these products are very dangerous also if the spillage happens or leakage happens or it comes from the industrial sites. So we need to remove it, and there are two different methods which can be used. One is the old conventional method, wherein we need to clean it manually, and that is time intensive also, and also it requires a lot of labor, and economically also it is not feasible. So if you see, like in India itself, from April to October 2023, we consumed almost 137 million metric ton of petroleum. So imagine that, this is just for 6 months in India, and if you consider the whole world, we are using these petroleum in a huge, amount. So if the spillage is happening, the leakage is happening, so it is contaminating our environment. So it can contaminate the soil, it can contaminate the water, and it needs to be handled in such a way that it is cost effective also. And one of the solution is using the microorganisms which are always present, but we need to find out, I mean, what microorganisms are actually good for this purpose. So bioremediation is one of the solution, and for that purpose, we selected. What we did actually, we got the samples from one of the locations where they store the petroleum and petroleum products. So sometimes, when you are transferring from the depot to the vehicle or things like that, so it gets leaked, or the soil gets contaminated with this leakage. So what we collected these samples from these different locations in India, from different depths, like from the top and then going down up to 1 meter. And from there, we identified by using these biochemical methods, we identified 5 potent microorganisms which can actually degrade petroleum products like petroleum hydrocarbons. So out of those 5, one of them gave us very good results. Like, it was able to degrade petroleum up to 90% in lab and microcosm settings. So that was our first publication. We already published in May 2025. And these microorganisms, how it is working actually. If the petroleum is there and the spillage is there, the contamination is there, this biodegradable these bacteria cannot work directly on it. So it has to be broken down to increase the surface area. And for that purpose, this bacteria is producing biosurfactant. So biosurfactant you can consider as, in your kitchens, if you have dishes with the greasy dishes, if you want to clean them, you use this detergent, right? So these biosurfactants are acting as a detergent, which is breaking down these uh, petroleum and into small missiles, which increases its surface area so that the bacteria can walk on it. And in our experiments, we found that it is drastically reducing the surface tension also, which is 27 And emulsification, like emulsifying it, we achieved around 85% emulsification index, and the critical missile concentration also, it was as low as 70 milligram per liter, indicates high efficacy. Once we identified this bacteria, we went for the whole genome sequencing of this bacteria, and we saw that, okay, this bacteria, not only it is degrading the hydrocarbons, petroleum hydrocarbons, but it is able to survive in that environment, the harsh environment, which is in the toxic conditions. It has a lot of toxins mixed in that environment. So the bacteria cannot survive. So they have to have a defense mechanism for that. So the bacteria was having this defense mechanism also. It can produce biosurfactant, which is very essential to act on this kind of spillage. And on top of that, this bacteria is able to degrade most of the petroleum compounds, like alkanes, petroleum PAH, and BTEX compounds. So to achieve this goal, what we did sequencing of this genome, the sequencing of this bacterial genome, and we tried not to use just the short read sequencing, which is Illumina. We added long read sequencing technology, which is third generation sequencing technology, to it. So why we use these two? Because
Thank you.
Oxford Nanopore, though it is giving us quite long reads, but it is full of errors, like 80 to 85, 90% is the accuracy rate. So if it is not good, like in terms of the quality or the accuracy, so we have to use a more the Illumina, like, which is more good in terms of the accuracy. So we try to use these two technologies together, and we used a hybrid sequencing approach for this, wherein we are going to take the advantage of these two technologies together. Uh, and so we used a unicycler, and we tested a lot of other softwares, but we got very good results with the unicycler, so which can take both the reads together, and we were able to assemble it in a very good manner, where we got only two contexts. So the N50 value, which tells you that how good your assembly is, it was around 5.54 MBB. And we got two circular, one circular chromosome and one single plasmid. And we then tested that how good this assembly is. So we used this Busco check M to check the quality of the assembly, and they use some of the key genes which should be there. If it is there, then you can say that, okay, the assembly is complete. So Busco gives us 100% confidence that it is complete assembly, and CheckM also, it gives us that 99.7, which is very high. So we are confident that whatever we have assembled is a good assembly. And after the assembly, we actually submitted the raw data as well as the assembled data to NCBI. And there was only one genome which was available for this particular species, and ours was the second one. And after the review from the NCBI, they considered this strain of Rhodococcus indonesiensis as the reference genome for this species. Now, how actually it is working. So these crude oil mixtures, I mean, they have three things. Like, one is alkanes, and the other one is polyaromatic hydrocarbons and BTEX compounds. So these all compounds uh, needs to be degraded when this bacteria is acting on. And most of the time, in a real situation, one bacteria is not able to do all these things. So there is a group of bunch of bacteria which are there in that environment, and they are helping each other by producing these enzymes, so that they can degrade these things. So what we found that not only this aerobic but also anaerobic degradation pathways were there in these microorganisms. So when the oxygen is there on the surface, so this oxidative or aerobic pathways were acting and then degrading it. And once it is deep inside this village, then it cannot work. So in that case, they were having a good set of genes which can actually degrade it in oxygen depleted areas also. So it was having both oxygen rich and oxygen depleted environment genes which can be used for the degradation of hydrocarbons. So if you see the genes which are involved and the pathways which are involved in the degradation of petroleum hydrocarbon, we were able to find many genes or the operons which are actually responsible for it. If you see that the oxidative cycle which requires monooxygenases, which is ALKB, which is well known, if it is there, then that means that this bacteria can degrade the hydrocarbons in oxygen or oxidative environments. So subterminal oxidation, long chain alkanes, and downstream processing, all these genes which are required from top to bottom to make it available for the TCA cycle. So it was there, and also they are having the genes which are responsible for this PAH and BTEX degradation. So in totality, this network of genes which are there in this single microorganism, so you will not find it. So this is something novel, and this bacteria is able to move from aerobic to anaerobic, anaerobic to aerobic, depending on the situations, and it can degrade the petroleum products or petroleum hydrocarbons in up to 90% of 90% it can finish it. So what are the biosurfactants pathways which are available in it? So there are multiple. So it's not just one biosurfactant, but there are many which can act on these petroleum hydrocarbons and make it available for the bacteria to digest it. So trihaloase lipid, mycolic acid pathway is there, phospholipid pathway is there, and lipopeptides are there. So we identified these genes by doing the first we did the structure annotation where we identified how many genes are there. And once we identified the genes, we got protein sequences which were used for the functional annotation. So we used many tools for the annotation and to see that if it is able to or it has a set of genes which can degrade hydrocarbons. So there are some based on previous studies, the databases are available which has set of genes which if you find it in your genome, that means that it has capacity to do the hydrocarbon degradation. So this is the method which was used. And if we talk about the stress conditions, so the bacteria is there in that environment, but it has to survive. So if it cannot survive, even though it is having all the mechanism to destroy the hydrocarbons, it cannot because it will die. So it has to survive in that harsh environment where you have oxidative stress. Sometimes, a lot of reactive oxygen species are there. Heavy metal toxicity is there in that environment, so which cannot be tolerated by the bacteria, they will die, and heavy metals and toxins are there. So, but this particular bacteria has a set of genes which make them survive in this environment and act on the petroleum hydrocarbons. So this I already mentioned, and we got this set of genes like superoxide dimutases, SORT-AC, which is actually helping them to fight in the oxidative stress conditions. Heavy metals, we got a set of operons basically, which is required to fight them against the heavy metal. And for the copper also, if the copper-rich environment is there, so they can survive in that environment. Other osmotic and thermal stress conditions also, we've got a lot of genes and proteins which are responsible to survive. So once we assembled this genome and did this functional and structural annotation and associating with whether it is going to degrade the hydrocarbons or not. Once we establish it, then we also try to find out that whether it is how it is sitting in the phylogenetic tree if we consider it. So for that purpose, we did species level comparison, like genome comparison, and genus level comparison also. So for species level comparison, we took the only available genome, which is CSLK0103, and I compared this with our genome. Rory is the software which is used for this purpose. And we also considered all other genomes in this genus. So genus level comparison also we did the genome comparison. And we found that it indeed belongs to this Rhodococcus genus. And this is a novel species under Indonesiensis. So you can see that DNA hybridization, this is digital one. We got 88.2% of similarity, and average nucleotide identity with the CSLK01 is 98.6. And we also did the amino acid identity, that how good they are, close to each other. So it was around 89%, close to 89%. So while comparing this with the already existing species of Indonesiensis, so we found that our strain is having 711 unique genes, which is making them unique for this biodegradation of petroleum products, which are not there in the other species of the same, other strain of the same species. So these unique 711 genes, basically, they are the one who are actually playing a role in the biodegradation. Uh, so So these I already mentioned, and we found that okay, these are part of these 711 genes which we actually identified. So that means though they are same species, but the strains are having very specific number of genes which are present only in SRG1 but not in the other strain. So it has the complete things which are required. That means to survive in the environment where actually it is present and which is toxic. It is not like that is going to die. So it has a machinery, it has a mechanism to survive in a toxic environment. And once it is surviving and then multiplying, so it is also able to access the hydrocarbons which are there in the environment to degrade it. For that, biosurfactant pathways which are required to act on these pollutants. So this biosurfactant is also there with it, and on top of it, like it can degrade also without taking help of the other bacteria in the environment. If they are there, they can produce some of the enzymes which are required. It's well and good, but if not, then on its own, it is able to degrade the products. So this I already mentioned that it can survive and then in catabolically, it is quite good to digest these hydrocarbons. So this particular work has been deposited in GenBank. You can see that the chromosome and plasmid is deposited under this project, bioproject ID. So if you want to access, you can go to NCPI and access these data sets. And also, if you want to do or repeat this work, you can do it in your lab because the raw data is also available under this project. And we have already published two articles with respect to this work in Nature Scientific Reports. And the first one was published in May, and the second one recently in December 2025. So with that, I want to conclude that, okay, this is what we have done at Banasari Vidyapeeth, and thank you very much for listening.
Thank you, thank you very much for this wonderful presentation. It's very interesting.
Thank you.
Thank you. Any question? Any question for this first conference in this day? Now we are going to move on the next conference of Dr. Amna Hariga. Are you here, Dr. Hariga? She comes from Tunisia. I don't found it in sinon, on va passer à la prochaine. Oussama, are you here?
Hi, Atiqa. Actually, Emma
Ah,
is connected, but can't share her.
okay,
She's
okay.
connected, but she cannot share her
Je-
screen because she's not host right now.
euh, elle lève la main, s'il te plaît, Amna, tu lèves la main pour que je puisse t'intégrer. J'arrive pas à te retrouver. Ah, ok. Welcome, Amna. Welcome. Now you can present Dr. Amna Hariga. She's research team leader. She's researcher team leader, assistant professor from Institut Pasteur, Tunisia. She's going to present a conference entitled Bind, an all-driven bioinformatics platform for computer aided drug discovery against infectious diseases. Amna, are you here? You can present?
Hi, can you hear me?
Yeah, I hear you, Amna. مرحبا, مرحبا.
Good morning, everyone. Sorry, I'm using someone else's computer and didn't realize that you can't see my name. I have
No
issues
problem.
sharing my screen
Yeah.
because it doesn't offer me the possibility to share my screen. It's sharing whiteboard. That's the only thing that I have. Can you handle that as a host? Maybe it's an option from your side.
No, I have another option.
Okay.
You have the same option for me
yes.
and you. Huh?
Okay. It doesn't allow me to share my screen. The only thing I have is a whiteboard. I never saw that in you only see a whiteboard, right?
Yeah. Just a minute, Amna. I want to resolve this problem. I don't understand.
It only gives me the possibility of sharing a whiteboard.
ONS. Okay. I don't understand. ONS.
Hello, Atika. Maybe I would have to present myself, to share my presentation. Meanwhile, Emna can resolve her sharing problems. What do you think?
Yeah. Oh, it's okay for me.
It's okay for you, Emna?
Yes, no problem.
Okay.
Okay. Thank you. Thank you.
If it doesn't work still on my computer,
You can send me. You can send me your presentation, and I will
yes, I can
present
do
it.
that, and someone like just make the slides move for me.
Okay. No problem. No problem. I can do it.
Okay.
You have the floor, Usama.
Okay. Thank you.
Usama, I can present you Dr. Usama Swawi, Associated Professor at the Higher Institute of Biotechnology of Bija, Researcher Associate at the Laboratory of Bioinformatics, Biomathematics, and Biostatistics at Institut Pasteur of Tunisia. He will a conference, emergence and evolution of a present conference entitled Emergence and the Evolution of Cricoides Transmitted Viruses in the Mediterranean region, spatio-temporal dynamics and epidemiological insight. You have the floor. J'ai un peu déformé votre présentation, je sais pas.
Pas de
You have
souci.
the floor. You have the floor.
Oh,
You have the floor.
thank you. Have you access to my full screen or not?
Yeah, it's
Okay.
clear. It's clear.
It's okay. Even now, it's okay. Because
Okay, it's okay. It's okay.
okay.
You can start. You
Thank you,
can
Atiqa.
start.
Okay, thank you, Atiqa. Thank you all. It's a pleasure for me to join this first conference, and I'm happy to see you again. I will give you some insights about my research regarding the emergence and evolution of glucoides transmitted viruses in our region, namely the Mediterranean region, by doing spatio-temporal dynamics and epidemiologic analysis. So I am a research assistant I am an associate professor of bioinformatics at the Higher Institute of Biotechnology of Beja, and also research associate at the Institut Pasteur of Tunis. So I will present right now two case studies. The first one is about the blue tongue virus. And This virus, the blue tongue virus, is an airborne virus considered as a major threat to global health economy. And it, it causes annually multi-million dollar losses for farmers or for all the agriculture economics. And Tunisia has experienced several BTV incursions. The first one that was reported dates back to the 90s. Okay, and our aim here is to better characterize the geographical origin and epidemiologic characteristics of these incursions in Tunisia, aiming at counteracting the new epidemics that may follow in the upcoming years. So, just to remind you, in these about the mode of transmission of this virus. Actually, the cycle, the viral cycle is the Chilochidus adult species bite infected ruminants. And after infecting these ruminants, it sucks blood and the virus replicates in this vector and can also be transmitted to other animal. And this is the main mode of transmission, but there are other modes like the oral one, the transplanted one, the venerable one, and so on. So, and this virus is composed of seven structural genes from VP1 to VP7 and four non-structural proteins NS1 to NS4. The outer capsid consisting of VP2 and VP5 may be considered as the best one of the genes that can give us insights about the phylogenetics and also the phylogeography of these viruses regarding its high level of mutation. And this is why we consider them as the ideal candidates. So, just to give you a brief insight about the methodology that we have adopted to conduct this project, we first collected data sets from the public repositories. We performed first a classical phylogenetic analysis, followed by Bayesian analysis, and finally, what we have inferred was statistically assessed by doing posterior probabilities, bias factor, and so on. And at the beginning, we have 76 sequences, from which we have 7 from Tunisia. We use DraxML, PhyML for phylogenetic analysis, BEAST for Bayesian analysis, and the statistical analysis were performed through the R package and also by BEAST itself. So what we have as main results here is that, as I said previously, the first cases were reported to be happening at the 90s, but our analysis showed that the TMRCA stands for Time for Most Recent Common Ancestor, revealed that the BTV, our virus, appeared first in Tunisia in 1966. Sorry. So, and this we have also noticed that there are some introduction of similar strains of BTV2 isolated in Tunisia in the 2000s. But we have also revealed the presence of other entries and other epidemics or local epidemics that occurred in 93. 2004 and also in 2009, and this is not what we have observed that really in on the pitch, because the only cases that were reported as epidemic in Tunisia were only reported in 19. So what we have noticed here that we have revealed that there are some other outbreaks that have been under radars for years. And this is what our analysis showed. We also, by trying to retrace the phylodynamics of these strains, we have, by doing this through a Google Earth panel, we have revealed that the root of all the data sets that we have already collected would be in Cyprus, that was followed by a transmission to South Africa. And during the 60s, these strains emerged in North Africa and other African countries like Cameroon and Nigeria. And only in the 70s, we have broadcasted a BTV in Tunisia, namely we have the BTV14 as serotype. Other BTV serotypes originated from Morocco, Nigeria, and also from occupied Palestine, and that spread in Tunisia in the 2000s, and with the two strains that are BTV6 and BTV3. What the question that raised about this is that we have two questions. The first one related to our neighborhood. First, because we are we are a neighbor of Algeria and Libya, and For us, we did not retrieve any transition from Tunisia to Algeria, and this is what was really surprising for us, given that we all know that there is a cattle movement and smuggling and trafficking across the borders of neighboring countries. And this result was really unexpected. Our explanation for this is that the low number of sequences that are from these countries, especially in Libya, we do not have sequences at all, and only few sequences that are not public in Algeria. This may explain why we do not reveal some interactions or some routes of transmission from Tunisia to Algeria or from Tunisia to Libya. The second thing that was unexpected for us, that we have revealed that some transition was between Tunisia and the occupied Palestine, and especially it was also surprising because we do not have at all commercial and trades and so on. But the explanation for this is that the wind currents could explain could contribute to the explaining of the circulation of the virus between the countries surrounding the Mediterranean basin. And this was already observed from Cyprus to Lebanon as well. So this might be an explanation for this transmission. So, regarding now the enter and the intra-serotype reassortment, we have revealed that BTV1, 2, 3, and BTVY subtypes are detected in Tunisia, came from several restocking and intra and inter-serotype. And most of the restocking have been taken place between the 1970 and 2000. So this analysis was published in 2 years ago with my colleagues, and now I will move to the second case study, which is the emergence and the evolution of episodic hemorrhagic disease. This disease is quite near the same. The virus share many things with the BTV virus, as they have the same hosts. The same panel of hosts, and also they are both of them are transmitted by the same lipocollocoides. So the vector is the same and the hosts are the same. So our aim here is to reveal what are the evolution patterns of the hemorrhagic diseased virus in the Mediterranean region as well. So this virus is also an arbovirus, and is also transmitted, as I said, to wild and domestic ruminants through collocoides. And since 2006, we observed high morbidity and mortality cases of EHD, okay, in Mediterranean region, especially for cattle and deer. And the temporal and geographical origins of these incursions remained unclear. So here we aimed to investigate the evolutionary history of EHDV in the Mediterranean region to highlight the epidemiological features and patterns and the relationships with genetic diversity. So what we have done, as for the other virus, we extracted from gene bank for the VP2 and VP5 segments that were isolated in the Mediterranean regions from the period 2006 to 2023, and blasted them to obtain a final data set of 68 and 91 nucleotide sequences. For these data sets, we conducted Bayesian phylogenetic analysis, and we inferred discrete models from geographical origin, serotype, and host. So what we have revealed as a result, there's the temporal origin analysis showed that the TMRCA for the VP2 route is estimated as 1,887. For VP5, the segments for our analysis estimated the TMRCA to 1,676. The spatial reconstruction indicated that the United States of America has the highest root state posterior probability for the VP2, and concerning the VP5, the highest root state posterior probability is shared equally between the USA and Japan. In other words, it means that the viruses that are now circulating in the regions have as origins mainly USA and Japan. So now while trying to revealing the patterns of geographical patterns, our analysis suggested that the EHDV started its circulation in the Mediterranean region by affecting first France and Tunisia after virus transmission from East Asia in the early 30s and North America in the 50s. However, for the VP2 transition route linking to USA and France is statistically valid, whereas for VP5 the route from Tunisia to from Japan to Tunisia was not significant. So, considering now the hosts for VP Sang, the virus transmission was extremely significant from cattle to deer, or from deer to cattle, with a bias factor of 28, which is very high. And also from Culicoides, the main vector, to cattle as well. We have a bias factor of 200. I just remind you that a bias factor higher than 3 means that the transmission is really highly probable. For VP2 segments, the highest bias factor was detected for deer to cattle, and the transmission route was significant also for Culicoides to cattle as well. So what we have, what I have as insights from these two analyses, is that we have described the evolutionary history of EHDV in the Mediterranean region, including Tunisia, Algeria, Morocco, France, Spain, and the Palestinian territories. We provided a strong foundation here to understand the phylogeography and geographic spreads of EHDV in the Mediterranean region. These may allow us to have better strategies for animal health monitoring, and factor such as vector and host distribution and ruminant rates and prevailing winds may have a major role in EHDV spread within the region, acting as intensifying factors for virus propagation. Future research that we have to conduct should focus on whole genome sequencing to enhance the detection of reassortment events and to refine the revolutionary insight, because as I said, we have only focused on the Mediterranean regions, but maybe by adding some other sequences originating from the surrounding area here may improve our analysis, and so we can have a better monitoring of the disease. And implementing an active surveillance routine of the virus in both wild and domestic ruminant is strongly recommended to identify new serotypes and observe the viral dynamics. That can be very crucial for all the agriculture system to have to lower the losses for farmers. So this article was also recently published in Frontiers in Veterinary Research. I would like to acknowledge my colleagues and friends, namely Professor Andrea Ali Ben Kahla, as a founder of laboratory, and also our team leader. My colleagues that strongly helped me in my research, namely Marwa Arabi, who is the postdoc in our lab, and also Emna Harika, that will be just talking after me, and our sponsor, like the BIND project, the Zodiac project, and also the African Bioinformatics Institute, for which I am a group leader as in this African framework. Thank you.
Thank you very much for this excellent presentation, wonderful presentation. And we pass the floor for the next. We are going to move on the next conference of Dr. Emna. Can you share your presentation, please, Emna? Are you here?
Yes, it's still not working. It's the only option I have is a white screen. So maybe Usama can put my slides, and I know he already seen this presentation. He will be probably trying to move the slides as I speak. Would that work for you, Usama? I don't know if he's hearing us.
Il est là, il est là. Perhaps it's a problem for compatibility, PC compatibility.
Yeah, maybe.
I don't know.
So I shared the slides with Usama. He will be trying to do that from his side.
Not problem.
Because I'm at another event and my battery died, so I borrowed my one of my
Okay.
students' laptop to do the
Okay.
presentation.
Okay, okay.
Sorry for that.
Oh, no.
Okay, so the slides are here.
Are you seeing my slides?
Yes.
Emna's
Yeah.
slides.
Yeah.
Okay. We share more than slides with Emna.
Yes, so my name is Emna Hriga-Swiai. And hi everyone. I'm very happy to be here and very sorry for the delay in presenting my work. So today I'll be presenting the use of artificial intelligence in the drug discovery and presenting some success stories and innovative approaches from my project called BIND. So I'm a researcher at Institut Pasteur, and next slide. Next slide. Usama? So yeah, so I'll be presenting the AI, and I'll be saying next slide every time I need to move. So AI is now invading our daily life, as everyone is conscious of that. And it appears in many type of applications that go from vocal assistance, photo editing, face recognition, content recommendation, etc. And if we draw a very simplistic but yet very accurate definition of AI, AI refers to development of computer systems or machines that can perform tasks that typically require human intelligence. So we have some examples. It can be understanding natural language, solving problems, recognizing patterns, making decisions, which is not part of the software applications as in a very classical way. So also we have a second part of this definition that was proposed by ChatGPT in October 2023. AI systems use algorithms and data to learn and improve their performance over time, allowing them to adapt to new situations and tasks. So here I would like to, I am underlining the most important things in this definition, which are the use of algorithms and data. So AI systems are not about algorithms only, it's also about data, and we'll see how we tackle this in our research. And the idea is to make these systems learn and improve as they adapt to new data and new situations. Next slide. So a basic way of looking at AI. Next slide. Okay, so AI is very different from classical programming by the fact, as we saw in the definition, by the fact that in classical programming, we do code hardly the rules for a system, and then we ask them using data to provide answers. In machine learning and AI, in a general manner, what we do is that we give data and we give corresponding answers, and we ask the machine to learn rules. So the rules are the machines and not ours, as in classical programming. And machine learning is the heart of the AI field. And we will see that deep learning is now what have made AI a very important technology of our era, is mainly the deep learning algorithms. Next slide, please. When we talk about a machine learning pipeline, most of the time we will have different steps that go from having a task, an information, data. It starts with data. So most of the time we will be building data sets, but these data sets are not intelligible for the machine learning or the AI system. So we need to process these data, and through the data pre-processing, we do what we call feature engineering. Next slide. These two steps are very important, and they prepare what comes after, which is choosing an appropriate technique from the machine learning or deep learning era in order to train these models, evaluate their performances, and then see if they are performant and accurate enough for deployment in real-life applications. Next step, please. So when we move from machine learning to deep learning, what happens is that the deep learning, in the machine learning, we prepare the data and we do this feature extraction as illustrated on this figure here. It's a very human-intensive task that used to be done by what we call machine learning engineers. So the idea is to transfer information. It can be biological data, chemical data, an image. Then we need to transfer that into basically a numerical matrix or a vector. And that's the only format that AI or machine learning tools would understand. Now, deep learning is an advanced technique that uses neural networks and that have a very deep architecture that will perform feature extraction by themselves. So the idea here is not to introduce a biased selection of features done by human, but let the machine learn the features themselves. The idea is to have the least bias from the humans as possible. So what are these neural networks? The neural networks are an abstraction of how the brain, the biological brain function. So it should mimic the human brain to achieve tasks, but remain a simplified and abstract representation of what is how the information is collected, treated, and used by the human neurons within the brain. So as I said, the deep learning approaches are meant to have a feature extraction part among them in order to not use the humans or not be dependent on the human knowledge to describe the data. Spoiler alert, we still need the humans even if we do deep learning approaches. Next slide, please. So there are different types of applications that we use in deep learning. Major types, and I'll go very quickly on this. We have CNNs, which are the convolutional neural networks that are very important techniques for computer vision, image analysis mainly. We have RNN and LSTM, which are the most used type of algorithms in treating text in natural language. And we also have graph convolution networks that are the most suitable into analyzing graphs, networks, and connection between individuals within society, for example. Next slide, please. So LLMs are the next generation after these, but most of the time would build upon some of these architectures in order to derive what we call now large language models like ChatGPTs and other type of generative AI. So if we move to successful application in the biomedical field, we have seen a lot of impactful applications, either in drug discovery with new molecules that have been conceived using AI. We have, of course, diagnosis applications using biomedical or clinical image processing for diagnosis. And we all have heard about the Nobel Prize that have been awarded to the DeepMind researchers for developing the AlphaFold. So these are different applications for different types of medical or biomedical types of data that are processed with different type of AI tools. Next slide. So what we do in our team, now I'll be more talking about our field of applications, which is the computer-aided drug discovery. Drug discovery is a very lengthy process that starts at the early steps of discovery, target validation, and lead identification, and that goes up to the latest phases of clinical trial in order for one molecule to reach the market. Most of the time it would take up to 20 years and $2 billion in order for one molecule to reach the market. So when we do computer-aided drug discovery, we introduce structural bioinformatics and recently AI into the pipeline in order to shorten this process. Next slide, please. But also to introduce or to leverage the outcomes. So what we do for infectious diseases of poverty and most of the time neglected tropical diseases. Next slide. We do both structural bioinformatics, AI into the pipeline. We introduce these techniques, but we also do what we call drug repurposing, which means that we provide new applications for drugs that already have reached the market, but we provide novel application of these drugs. The idea is to lower the cost. So the idea is to lower the cost of the pipeline. Yeah. Yes, we lost the slides.
Just a minute. Perhaps we have a problem to connect with us. On peut l'attendre bien. Qu'est-ce qu'on doit faire?
I also shared the slides with you, Atika.
In email?
Yes, your personal email, yahoo.fr.
Okay.
So maybe he has connection issues.
Okay. You can resend me because it is a Google slide, no?
Yes, it's a Google slide.
In Yahoo, I can't
okay.
open And I send you now
Yes,
permission.
so I put share. Can you try now with your Gmail?
Yeah, I
Okay.
can. I
So
can.
can you skip to slide 15
Okay, okay.
to keep the time correct?
Okay, okay. No problem. You're welcome.
Okay.
This one?
So slide 15.
15. Okay.
Yes. Yes, that one.
Okay.
So I'll go directly to present what we do. So here I'm presenting our funding sources. So we are a multidisciplinary team based in Institut Pasteur of Tunis, and we have started working on introducing AI and deep learning in our drug discovery approaches since 2017. And next slide. So as part of a program called Arise from the African Academies of Science, we have proposed the BIND project, Bioinformatics and Artificial Intelligence for Drug Discovery. And it's a project, next slide, please, that is articulated in two main sub-objectives. The first one is apply drug discovery for infectious diseases. The second one is developing new approaches in drug discovery. So what we do is that our main objectives are, you have the diaporama button just at the
I don't
à
find
ta
it.
droite tout en haut. À
Here?
côté de partager, il y a diaporama.
Ah oui.
Juste à côté.
Ah, c'est bon,
Voilà.
merci. Merci.
T'en prie. So we have three main objectives. First, we have worked a lot on preparing good data sets for the approaches that we are developing based on the different infectious diseases that we work on. Then we have developed different machine learning and deep learning based frameworks for drug discovery. And finally, we also do the experimental work and we work up to validating novel anti-pathogen drug candidates. Next slide, please. So as we recall the machine learning based drug discovery pipeline, what we have is that we work on collecting, cleaning data sets related to the diseases that we're interested in. We do a lot of optimization of the encoding systems as part of how we improve the literature for AI for drug discovery. We train and validate either state-of-the-art models, but also we have developed new algorithms in our team. And finally, whatever comes from there, especially when we predict new biological activities of these molecules, goes into in vitro experiments, then preclinical studies, and hopefully we will have some molecules that will reach the clinical studies. Next slide, please. And then next slide. So first things first is the data. And we tackled that with CDLZB, which is an open source platform for anti-pathogen computer-aided drug discovery research. This is a database in which we have collected data from different other databases. We have curated and cleaned the data, but also we have enriched existing databases with data from literature. So we have been doing extensive literature review in order to collect two data sets. Next slide. One for leishmaniasis, the second one for coronaviruses, and we have enriched the existing databases with up to 4,000 molecules for each of the diseases. And we saw that, next slide, please, using these enrichment data points from the literature, we have been able to enhance significantly all different types of machine learning and deep learning algorithm performances, as you may see in the red square. So that's the impact of the added information from the literature on the models. So this research have demonstrated to which extent the preparation of the data sets in comparison to just using generic data sets from the databases would look like in terms of performance. Next slide, please. And then next slide. So as we have these data points, data sets, but also we have the models that have been optimized on the different pathogens, we went and deployed these outcomes into a platform that is accessible through the link that you see here. And what we have is that an open source platform that can be used by everyone that is interested in leishmaniasis and coronaviruses. So you can browse the literature information that we have collected with IC50 values, the chemical structure, etc., etc. But also we have a second panel, which is the prediction part. And that people can use directly the optimized machine learning and deep learning models that we have prepared in order to predict the biological activities of their molecules. Next slide, please. So basically, CDOS-DB is a platform that helps seamless access to AI-based predictive models for drug discovery. For now, it contains leishmaniasis and coronaviruses, but we are working into including up to 4 additional pathogens into the platform. This work have been published in 2024 and have been awarded the National Award for the Best Feminine Research for the year 2025. And I would like to acknowledge all my colleagues with their names presented on this slide. Next slide, please. So as we develop the AI pipelines, we also translate that into real discoveries. So what we do is that using the different approaches that we have been developing, I didn't present everything, but we have different approaches into the pipe, and we use them to identify 71 active molecules with 80% confidence in being active against leishmaniasis, which is a parasitic disease that we are interested in. So out of these, we have 27 that have been validated by the literature, as we identify that there are work that prove they are anti-parasitic, anti-leishmania. Out of the remaining, we have tested almost all the remaining molecules, and out of 40 molecules tested, we have 24 molecules that have validated effect. Next slide, please. And out of the 24, we have performed different type of in vitro experiments and identified 5 promising drug candidates that are now into preclinical studies under assessment and also being studied for patent filing for now. Next slide, please. So here I'm just showing one of the papers that have really went using machine learning up to the experimental validation. And for one paper, we have 3 new hits molecules that have been published in 2024 by Dr. Rafeh Walha. And the remaining, next slide, please, are into the preclinical studies currently. We have different work that is being into the pipe and different manuscripts under preparation with both my colleagues Rafeh Walha and Yusuf Zina Abdelkrim. Next slide, please. So to conclude my talk, I hope I wasn't very long. Through the BIND project, we have achieved different things. So we have all the aspects related to innovative methodologies. I presented the CEDAL-DB platform, but we have different methodologies that have been published, like Keros, Morgan QRL, and we also have different other new things that are coming for publishing soon, like the Cosmo and the Parafusion Net frameworks. We have 5 very promising anti-leishmania drug candidates that are into the preclinical studies currently. But we also did a lot of capacity building through training PhD students, postdoctoral fellows. We have engineers and research assistants that are in the lab doing the work. And we also have undergrad and master students each year joining the group. Next slide, please. So with that, I would like to acknowledge the whole team behind the research that I showed. So, namely Professor Akram Gizaini, head of our team in the laboratory. We have Osama Swiai that spoke just before I did. My colleague Zina Abdelkarim and Rafeh Welha, both senior researcher in biology, and all the young faces and team members that you can see that are mostly students, master students, research assistants, or PhD students in the lab. And thank you for listening.
Thank you so much for this presentation, substantive work. You did a good job, Amna.
Thank you.
Any question, please, for the audience? Yeah, Walid?
Yeah, hi. Thank you for both of them. I would have a question for Dr. Osama, if possible. I think he's not here. I think he's not here. Yeah, I think he didn't come back. Yeah, it's okay. It's okay. Then I will.
You can. He can ask you, Amna.
Of course, I'm open to on Osama's work.
Yes.
Respond to some things, but yes, let's try.
Okay, it's about the propagation of the virus he mentioned. He said that there were no known propagation into Algeria and Libya, and he explained the propagation into the Palestine and I mean the Middle East by the wind. So I was wondering why the wind didn't work on the east side, but went so far.
Yes.
So
no, this is a very valid question. You would say why it didn't work the other way.
yeah.
Actually, I think the problem for Algeria and Libya is the lack of data. So if we had more data, I'm trying, this is what I recall from the discussion as we were revising the manuscript, because that was one of the major concerns of one of the reviewer. And it's basically because there are sufficient data for Tunisia, and then we have sufficient data from the occupied territories and Palestine, but too few data points for Algeria and Libya, especially for Libya, I think, which means that the relevance of the connections are not very strong. And this is why it seems like it's a weak connection.
Okay.
So
even though if we take into account the smuggling activities, because if I'm not wrong, you may correct me, the smuggling is from Algeria to Tunisia, right? Not the other way around.
I can't recall that information, sorry.
Okay,
been more sort of an external collaborator on that work, so
all right, thank you.
I don't have all the details. Yes.
Yeah. Thank you, Amna. Thanks again.
Welcome.
Yeah, you can ask a question.
Hello. Yes, good morning. Can you hear me?
Yes.
Dr. Belgesem Farah Fadwa. Yeah.
Yes. Can you hear me? I have a question about this current topic. So basically, thank you so much for this great work. It looks really interesting for me, especially for someone who is interested in bioinformatics. This is really, it sounds good. I have just a question about the database that you have made already. The application is for anti-pathogen drug discovery. That's it? Am I right?
Yes.
So, based on your experience, can we use your data just for this application, or can be used for somehow similar applications? Like if we are just looking for like novel enzymes for maybe industry usage, are we still able to use your data, or is just preserved for pathogens?
Good question. As I mentioned, this is why I show in the very first slide the definition of what is an AI system. The AI system is both the data and the algorithm. So you cannot dissociate these two components, which means that if your data set is only revealing information about an activity against one pathogen, it can be multiple pathogens at the same time, that your machine learning or deep learning algorithms will only learn that. That's the only thing that they are able to do. So if you would like to do additional applications, what is needed is basically have a data set that is serving that type of applications. Like, for example, if you have different enzymes, it can be in agroalimentary applications that you want to either enhance their activity or like inhibit their activity in order to have a certain outcome. You will need to have compounds that act on these type of enzymes. And then you'll be able to identify new molecules that will help you, act on your enzymes of interest. So it's basically, if you have the new data set, then transferring the machine learning and deep learning applications it can be very easy, depending on how easy for you to code. Knowing that all our publications are, we do open data, open source publications. So most of our code is also open on my GitHub or my students' GitHub accounts, if that helps.
Okay, thank you so much. It sounds good. Thank you.
Professeur Shelgi Hamedi.
Oui, bonjour, docteur Daïkra.
Bonjour, bonjour.
Je vous remercie. Juste une question pour le docteur Oussama Souaï, si vous permettez. Il
Oui,
est là?
non, il n'est pas là, mais Amna, vous pouvez répondre à sa question? Si
Ça
vous
dépend,
pouvez, bien
je
sûr.
peux essayer.
Oui.
Ok, merci bien. Alors, ma question, c'est comment les changements génétiques des virus transmis par les culicoïdes influencent-ils leur dangerosité? D'une part, est-ce qu'ils gardent aussi la même compétence vectorielle? Et ma troisième question, est-ce qu'on peut les surveiller et les contrôler? Voilà mes trois questions. Sinon, je les envoie vers le chat, c'est ça?
Alors, je préfère que vous lui envoyiez la question. Comme ça, je dis pas de bêtises parce que je suis pas du tout virologiste. Donc, tout ce qui est échange génétique, etc., je ne peux pas vraiment me prononcer. J'ai une petite idée, mais je préfère ne pas répondre.
Ok,
Je ne
merci
suis pas
bien.
très sûre de comment répondre à ça. Donc, vaut mieux lui écrire par email ou quelque chose comme ça et il va vous répondre en fait.
Ok, merci bien. Merci.
Compris.
Bonjour. Bonjour, docteur
Bonjour,
Idekra. Bonjour, chers collègues.
bonjour, professeur Chérif. Vous voulez poser une question?
En fait, je voulais remercier vivement et féliciter Madame Emma pour son travail. Ça m'a vraiment intéressée parce qu'on a appris beaucoup de choses. C'est vrai que son collègue, le précédent, avait parlé beaucoup plus de cadre épidémiologique, mais elle, c'est l'application de l'intelligence artificielle dans les domaines des drogues ou molécules. Et donc, c'est intéressant, c'est très intéressant. Mais je voulais juste lui dire, comment voyez-vous l'avenir de l'application de l'intelligence artificielle dans tout ce qui est biomédical? Est-ce qu'on va vers quelque chose d'extraordinaire ou ça va juste nous aider dans la découverte, dans les interprétations de certaines molécules bioactives? Donc, comment elle voit vraiment l'application de l'intelligence artificielle dans les années futures, d'après son expérience? Voilà.
Oui, c'est vraiment une question qui est, c'est vraiment le cœur de la discussion autour de l'IA maintenant.
Oui,
Alors, l'IA, avec toutes les technologies qui sont en train d'être développées, mais surtout aussi la capacité à avoir des données de plus en plus au format digital et de bonne qualité. C'est un peu ce qui fait la force des pays très développés afin
oui,
d'avancer, d'avoir des systèmes d'aide au diagnostic, des systèmes de découverte de nouvelles molécules, des systèmes qui comprennent un peu ou qui vont identifier des connexions au sein d'un génome, comprendre un peu plus la génétique, etc. Donc les applications sont diverses et réellement, c'est
diversion.
un outil qui peut transformer la recherche et les applications médicales en général. Maintenant, je pense que dans nos pays respectifs, les pays du Sud en général, je pense que si le bottleneck, c'est-à-dire là où ça fait mal et là où on risque
Mhm,
d'être très en retard par rapport à d'autres pays, c'est la partie données et beaucoup moins la
oui,
partie technologie. C'est-à-dire avoir des ingénieurs et des personnes qui peuvent développer de l'IA. Ça, on va pouvoir, on y arrive, on a énormément de gens formés, il y a les universités, maintenant tout est ouvert, les gens se forment un peu partout, même en ligne. Et je pense que si la personne, elle est capable et elle étudie bien et elle a les compétences, la partie technique, on est capable de l'aborder au même rythme que d'autres pays.
oui,
Mais les applications ne vont devenir relevant dans nos systèmes de santé et dans notre recherche que si on met l'effort nécessaire et l'énergie nécessaire pour collecter nos données, pour nos pathologies, pour nos patients, pour les maladies qui sont en train de sévir dans nos pays, etc. Et faire en sorte qu'on a des bases de données bien nettoyées et bien soutenues. Et là, par exemple, quand on parle médical, on a besoin de biologistes qui vont faire l'effort de collecter des données, de discuter avec des informaticiens pour savoir c'est quoi des vraies données pour l'IA. Donc,
oui,
la notion de données n'est pas la même pour une personne de l'informatique et une personne de la biologie. Et donc, cette discussion doit avoir lieu. Les médecins aussi, on travaille avec des médecins et on a du mal à avoir des médecins qui se motivent pour, par exemple, ils ont des données, mais il faut les annoter, il faut les rendre des données que nous, on pourrait, de notre côté,
utiliser.
du côté de l'IA, on va pouvoir les utiliser pour des
Oui,
diagnostics, pour des
oui.
compréhensions, etc. Donc, je pense que ça va être génial, mais si nous, dans nos pays, on a envie de suivre le rythme, il faut avoir nos propres données. Voilà.
Parfait. Merci. En tout cas, vous me donnez vraiment une autre, un point positif pour l'intelligence artificielle et ses multiples applications. En tout cas, je vous souhaite vraiment un grand succès par rapport à tout ce que vous avez fait comme travail. Je pense que c'est une petite partie de ce que vous faites en ce moment que vous venez de présenter. Donc, je vous souhaite une bonne continuité. Bravo.
Merci.
Walid,
I'm not.
Dr.
Sorry,
Benak.
it's okay. I'm back again. I'm sorry to bother you. Yeah, I just was wondering what kind of setup do you have in the lab, and I mean in hardware, in terms of hardware, and if you would be keen for collaboration, because what we lack here, it's really the hardware.
Okay, so the hardware is in Institut we have an HPC cluster, but it doesn't help that much in what we do because we need to have GPUs and TPUs, which is not there yet. So through the Bind project, we are now trying to acquire a server with a very powerful GPU, which is very needed to train deep learning models. So what we do is sometimes we collaborate, sometimes we put that on CPUs and it takes a lot of time. So the hardware part is a struggle, but for now we managed to do that. Sometimes we also use platforms like Kaggle or Colab in order to do some stuff.
Yeah, but that's not really
Depends.
sustainable, right?
Sorry?
That's not really sustainable. I mean, the cost in the long term is.
Yes, it's not sustainable, and for very long simulations, sometimes we end up using all the credits and we don't finalize the simulations.
Yeah.
Yes.
Yeah, I do have
The
that.
hardware is, I am working on putting a new infrastructure in place, but it's taken a lot of time to acquire the hardware and etc. So we are waiting for something to be delivered, hopefully
Well,
soon.
yeah, hopefully soon. So maybe in the future we may collaborate again.
For collaboration, if it's really relevant and we can help, of course, we are very open to collaboration. So
Yeah.
just
Sure,
write me an email and we can discuss.
Sure.
Whatever. Yes, sure.
Thanks again, Emma.
Thank you.
Voilà, on te remercie, Emna.
Merci
Et,
beaucoup, Atika. It
à
was my
toi.
pleasure.
Voilà, c'est un plaisir pour nous de vous revoir encore une autre fois, que ce soit pour toi ou Sama. Donc, c'est un plaisir d'avoir assisté aujourd'hui à ces deux conférences très intéressantes. Ça reflète le travail de fond que vous avez accompli. Donc, c'est vraiment impressionnant. La même chose pour la conférence de notre invité, la première conférence. Malheureusement, il a quitté la salle. Donc, je demande maintenant, on va passer au plaisir de vous revoir une autre fois. Vous pouvez rester avec nous, hein. Donc, je passe aux prochaines conférences. Donc, j'appelle Dr. Shelby. If you are here, just raise your hand. Dr. Shelby, are you here? I think no. On invite, we invite Dr. We jump in the second conference. Now we invite Dr. Jida Yed to join us. You are here?
Yes, I'm here. Bonjour.
Bonjour, bonjour.
Bonjour à toutes et à tous et merci beaucoup, Madame Atika, pour cette invitation. Je suis présente.
C'est moi qui vous remercie. Nous vous
C'est un peu
remercions.
serré pour moi ce matin. Je n'ai pas arrêté de courir, mais le plus important est que je sois là.
Merci, merci pour votre présence. Merci.
Avec un grand plaisir, Madame.
Alors, vous venez donc de France. Hein, vous êtes
Oui,
algérienne.
je suis algérienne. Oui, j'ai fait mes études en Algérie. J'ai fait l'INA d'Al-Harrach à Alger.
Voilà,
Oui,
et vous êtes l'une des compétences comme les Tunisiens, comme les Algériens,
totalement. Oui,
voilà.
totalement. Et
Donc,
c'est
c'est
un
un
honneur.
plaisir.
Oui, 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 c'est totalement, je suis totalement d'accord avec vous, madame. Et franchement, félicitations pour cette organisation. Moi, je suis
Je vous en
toute
prie.
oui pour multiplier et pour développer ce genre de manifestations et nous réunir comme ça de temps à autre pour partager un petit peu nos savoirs et les travaux sur lesquels on travaille et puis s'enrichir. Hein. Donc, il n'y a pas que nos étudiants ou nos élèves, même nous, les chercheurs. Donc, c'est toujours bénéfique un peu de partager un peu ce qu'on fait et puis collaborer, pourquoi pas, et développer un petit peu aussi ce qui se fait chez nous localement, notamment en Algérie, en Tunisie, au Maghreb, et voilà.
Très bien. Donc, je vous donne la parole pour présenter d'abord votre conférence qui
Oui,
s'intitule Non-Descriptive Early Detection and Prediction of Pre- and Post-Harvest Plant Disease
yes,
using Multimodal Low-Cost Sensors, Slot, and Machine Learning. C'est ça?
yes. It's a
You
great
have the
pleasure for
floor.
me. Yeah, but j'ai pas la main, je pense, pour partager. Je peux parler, mais je peux pas
Attendez.
partager.
Ah,
Est-ce
normalement,
que
je
vous pouvez
vous ai...
vérifier?
Oui, normalement, je vous ai
Mais-
donné... Ah, voilà, très bien. Vous pouvez maintenant partager votre conférence, votre présentation en cliquant sur le bouton vert en bas. Green button. Click just on the green button.
I will do it. Just one moment. I will share my screen. It will be okay. I think that it's okay for everyone. Do you see
Yeah.
my screen? Is it okay for everyone?
It's okay, it's okay.
Yes. So, good morning, everyone. I'm here. I'm so happy to be here and so happy to share with you. I can activate also my camera.
It's okay. You can, I can see you. I can see you.
Okay. So, thank you a lot. I'm really thankful for this invitation, and it's really a great moment for me to share some of my work with you. So, first of all, let me introduce myself. I'm an agronomist by training, graduated from the National Agronomic Institute of Al-Harrach in Algiers, with a specialization in plant pathology. For several years, I worked on fungal plant diseases, especially early blight. 3 years ago, I joined ISAN West in Brest at France, an engineering digital school as an associate professor and head of agronomy and ecology training. As we know, everyone, the major agriculture challenge today actually is to know how to use advanced sensing technologies and data-driven approaches can support decision-making in crop production. For example, I investigated hyperspectral imaging to monitor nitrogen nutrition in potato crops. Also, I tested chlorophyll fluorescence analysis using the multiplex 330 sensor for early diseases prediction. And I'm currently working on the development of mobile application integrating physiological and environmental data. So let me share some of my work, such as non-destructive early detection and prediction of pre- and post-harvest plant diseases using multimodal low-cost sensors, IoT, and machine learning. As we know too well, fungal diseases destroy up to 40% of global crop production every year. In Nigeria, as in many parts of North Africa, post-harvest losses in potato can easily reach 30 to 50%. For small farmers and cooperative, an outbreak of early blight or late blight can wipe out an entire growing season. And once symptoms become visible, it's often too late to optimally limit the damage. So the real challenge today is the anticipation. Traditionally, in plant pathology, we collect a leaf or a tuber, send it to the laboratory and wait for the results. Is it right? Yes. This approach is destructive, time-consuming, costly, and impossible to deploy in real time over large areas or for smaller farmers. As a result, treatments are either applied too late or used preventively across entire fields, increasing costs and environment impact. We clearly need non-destructive, real-time and rapid, and accessible diagnosis tools. Today, fully non-destructive technologies and are within reach. In my work, for example, I tested three main approaches. The first one is hyperspectral imaging for monitoring nitrogen fertilization. The second one is around multiplexed chlorophyll fluorescence for prediction early blight. And the third one is an application currently under development based on a very low power IoT using LoRa and 4G. All components are assembled from commercial available of pre- and post-harvest diseases. All these, tools share the same philosophy, non-destructive, field deployable, and accessible. I start with the first approach regarding hyperspectral imaging, analysis leaf reflectance across more than 100 wavelengths between 400 and 800 nanometer, allowing the estimation of chlorophyll content and the identification of spectral signature related to plant nitrogen status. The acquisitions are performed under white light and UV excitation, the latter enabling the analysis of chlorophyll fluorescence, especially the red to infrared ratio. It's the key indicator of nitrogen status. So, as we know, everyone, more nitrogen is not better for our plants. The experiment was conducted on potato using 4 nitrogen treatments, 2.0 to 2.3, ranging from no nitrogen input to 3 split application to tallinn 150 kilogram by hectare with 3 replication per treatment. Results showed a higher absorption and fluorescence level for T2 and T3 compared to T0 and T1, consistent with nitrogen supply. However, a spectral response for T2 and T3 were very similar, suggesting that the additional nitrogen applied in T3 did not provide a significant physiological benefit or need. Thus, preliminary results indicate that intermediate nitrogen dose may be sufficient, opening the way to fertilization optimization with both economic benefit and reduced environment impact. So spectral data suggests that the third nitrogen application is not necessary. We move to the second approach around chlorophyll fluorescence, which is one of the earliest indicators of plant stress. When a pathogen attacks a plant, photosynthesis is disturbed long before necrosis or sporulation becomes visible. The multiplex 330 is mobile, UV visible, autonomous, and non-destructive fluorimeter designed to measure real-time physiological parameters of plant on leaf or on fruits or seeds. So fluorescence allows us to detect stress days or weeks before visible diseases. Okay, you will say to me, okay, Gida, everything is okay. So maybe you will ask me the question around how it works. So I will answer you. It's so easy. We put the device at 3 to 4 inches from the leaf. We press the button and the magic affair. Based on plant fluorescence, the instrument excites plant tissue with specific light and measures the emitted signals to calculate easily physiological indices. For example, we can see about chlorophyll, its ratio between far-red and infrared fluorescence ratio based on chlorophyll reabsorption. We can also flow polyphenols, which is measured with, via the screening effect of polyphenol on exciting light by comparing a reference light to a specific light, and the blue fluorescent component, which is an indicator or detected under UV excitation regarding the phytoalexin, which plant defenses. It's produced in response to biotic, mainly biotic stresses such as fungal infections. So what about plant fluorescence? We know everyone that all objects absorb and reflect light differently. For example, plants absorb and reflect light also. we can say that plants absorb or drink light like we humans drink water, especially red, green, and blue light from the sun. Okay, but they don't keep it all. A tiny bit of this light is reflected as invisible colored, and that's what we call fluorescence. This reflected light changes if the plant is sick, tired, or well-fed, like a health thermometer for us humans. Fluorescence is in plants. It represents plant. Plant fluorescence is the natural dashboard of plants. Chlorophyll emits red light when all is well, as we see in this picture. But when pathogens attack, the plant triggers the blue alarm before a spot appears. The multiplex reads 3 signals in less than 1 second without touching the plant. Results early detection, like a smoke detector before fire. For example, we have blue-green fluorescence, which it raises when the plant fights back in case of phytoalexin. The red fluorescence higher when everything is well. It's an indice of a healthier chlorophyll. And infrared shows overall plant performance. Every these indices are measured in less than 1 second without harming the leaf. Let me explain more about plant fluorescence in 5 points. The first one, it's a health light naturally emitted, like an ion sign that lights up when everything is fine. The second point, it measures the plant engine. The green leaf equal engine running smoothly, and thick leaf equal engine stally. The third point, it detects early warning signals. It's like I explained it before, like a smoke detector before a fire. The fourth point, the multiplex instrument read 3 case colors or 3 signals. The blue regarding defenses against diseases, the red chlorophyll health, and the infrared overall energy. 3 dashboard lights to diagnose the plant. And the 5th point is regarding the advantages: fast, non-destructive, and rapid in real time measurement in less than 1 second in field, non-invasive, like an infrared thermometer for humans. As a result, we see that fluorescence measure early physiological response, changes in chlorophyll, decrease in ratio red fluorescence, infrared fluorescence with photosynthetic damage, accumulation of defense compound, increase in the ratio blue-green fluorescence, flavanol for phenolic under biotic races. Let's move to the third approach on what I'm currently working regarding IoT for pre and post-harvest monitoring. This ongoing application project relies on LoRa communication for areas without network coverage and 4G where available. Data are transmitted directly to simple mobile application accessible or application accessed by the farmers. The system sends precise alerts such as the north field high early blight risk in 8 days, for example. So the farmer can then treat only the affected area at the right time with the appreciate product. So we can treat only when and where needed. The fourth point is regarding AI for production, not a big AI, but smart AI adapted to the field. Raw data are fused using lightweight AI models such as random forest, XGBoost, and small CNNs for image analysis. This step is currently under development. The objective is to produce robust, explainable, and energy efficient AI models suitable for real-world agriculture conditions. As a result, on potato, under controlled conditions, detection occurs on 10 to 12 days before visible symptom. We have a ratio between a high percentage of sensitivity and specificity. Field trials are currently ongoing. The objective is to reduce, especially and mainly reduce the use of chemical treatment, leading to lower costs and reduced environment impact. As a conclusion, I can say that these technologies change plant disease diagnosis from reaction to anticipation, combining biology, sensors, and AI for sustainable agriculture. So we can say that biology plus sensors plus AI equal a new way to protect crops that is earlier, more sustainable, and smarter. These studies open the way to sustainable diagnosis. Thank you so much for your attention. Don't hesitate to ask me whatever you want of a question, and we can keep in touch. You have my email address. Thank you. Merci beaucoup pour votre attention.
Merci, Dida. Thank you so much.
You're welcome.
It's very interesting. It's very, very interesting. Any question, please? Des questions, s'il vous plaît, avant de passer à la prochaine conférence?
Je ne vais pas avoir beaucoup de temps, mais je suis preneuse de vos questions. N'hésitez pas. Et si jamais je vous laisse mon adresse email, n'hésitez pas à m'écrire. Moi-même, je suis en train d'explorer parce que moi, je suis agronome de formation, je suis biologiste, je faisais de la biotechnologie avant. Et
alors, merci beaucoup. Vous avez deux questions. Vous avez
oui.
Dr Belag Walid. On va commencer par Belag.
Ouais. Merci beaucoup. Merci beaucoup
Bonjour,
pour la présentation.
Walid.
Bonjour.
Bonjour.
Merci encore pour la présentation.
Toi.
J'aurais juste une question par rapport au coût. Est-ce que vous pensez que l'utilisation des sensors est meilleure en termes de coût que l'utilisation de drones, par exemple, où on peut c'est-à-dire prendre des images en temps réel des plantes et puis analyser les images plutôt?
Très, très bonne question, Walid. Alors, pour le coup, je pense que ces technologies sont de plus en plus accessibles. Ce n'est plus comme avant, hein. Donc, du low cost, on peut faire beaucoup, beaucoup de solutions avec que du low cost. Alors, pour tout te dire, moi, je suis, excusez-moi, moi, je tutoie tout le monde, hein, donc je suis
C'est pas
prof.
grave, bien sûr.
Merci Walid. Donc moi, avec mes élèves, on fait vraiment des solutions avec du low cost, des capteurs qui sont, alors on a plusieurs capteurs qui sont autour de 10 euros. Euh, voilà, à partir de 6 jusqu'à 10 euros, on peut avoir les capteurs. Et franchement, ça va tellement vite que par exemple, l'imagerie hyperspectrale, moi, quand j'avais fait mon premier test, là, je vous ai fait juste un échantillon pour participer à cet événement. J'espère dans d'autres événements, Inch'Allah, on aura autre chose à présenter. Mais il y a 3 ans, donc l'imagerie hyperspectrale, elle était presque inaccessible. Et ben, ça fait que 3 ans, et ben, tout le monde peut faire de l'imagerie hyperspectrale. Maintenant, je peux même développer donc avec mon mobile. Je travaille sur un projet étudiant pour les quatrièmes années, les niveaux quatrième année, et on fait des applications rien qu'avec l'image de l'iPhone, quoi, du mobile. Donc, vraiment, c'est vraiment accessible de nos jours, hein. Voilà.
Enfin, je pense, enfin, je fais cette réflexion, surtout, je sais pas, vous êtes au courant, mais les 2 dernières semaines, il y a eu des changements un peu en Algérie, surtout pour l'utilisation des drones. Il y a eu des nouvelles règles pour justement permettre aux gens d'acquérir des drones dans différentes applications, bien sûr. Et c'est enfin, je pense que ça pourrait changer un peu l'agriculture.
Oui, totalement. Donc, pour les drones, c'est vrai qu'il y a des règles. Par exemple,
Oui,
moi, je suis sur Brest, c'est une zone militaire.
oui,
Et je n'ai vraiment pas, c'est très, très serré, c'est très tendu, c'est presque interdit d'utiliser le drone. On a vraiment, oh là là, c'est un vrai casse-tête pour faire des tests avec le drone, parce qu'il y a une réglementation, et surtout avec ce qui se passe en ce moment dans le monde, hein,
oui.
les menaces, les drones, voilà, je te laisse imaginer. Mais en tout cas, c'est une technologie vraiment de plus en plus accessible.
Ouais, ouais. Mais une autre réflexion, c'est surtout par rapport à la facilité d'utilisation. Parce que à la fin, on voudrait bien que c'est l'agriculteur lui-même, pas l'agronome qui utilise
Oui, oui, oui,
au
oui.
jour le jour. Donc, qu'est-ce qui serait plus facile? Est-ce que c'est les capteurs ou les drones pour un agriculteur lambda?
Alors, pour un agriculteur lambda, il faut d'abord lui faire une petite formation. Comment il
D'accord.
utilise justement, oui, il faut le former d'abord pour la connexion parce que c'est pas toutes les connexions. Il peut pas utiliser n'importe quelle connexion. Il peut pas aussi, il faut le former aussi sur l'utilisation du mobile. Comment
Ouais,
justement il peut récupérer la donnée. Parce qu'en fait, le capteur, il va aller collecter la donnée, mais
ouais,
comment récupérer la donnée sur son ordinateur ou sur son téléphone portable et comment gérer cette donnée-là.
ouais,
Donc, il faut lui faire une formation. Personnellement, quand je suis arrivée la première année à l'ISAN, j'ai travaillé sur une formation continue, une petite formation destinée pour les agriculteurs, mais surtout sur comment utiliser, quelle connexion utiliser et comment gérer, justement, récupérer la donnée et la gérer sur son ordinateur.
très bien, très bien.
C'est-à-dire un agriculteur qui n'a pas une formation, il ne peut pas gérer
Ouais, ça va de soi, oui,
ce genre de solution. Ouais.
exactement. Très bien. Merci encore, merci beaucoup.
Merci
Alors,
Walid.
Merci.
merci Walid, merci Dida. Donc, il y a une autre question de la part du professeur Hamidi Chergif est là.
Oui, bonjour Fella.
Alors, bonjour, docteur Hayet. Alors, je vous remercie
Bonjour.
pour cette présentation très intéressante.
Merci beaucoup.
Quand vous l'avez bien dit, c'est comme un thermomètre.
Totalement, oui.
Ce qui m'a intéressé le plus et ce qui m'a attiré. Juste une petite question. Est-ce que ce thermomètre, on peut l'utiliser sur toutes les plantes? Ma
Oui,
première question. Et pour ma
oui.
deuxième question, comment peut-on détecter tôt les maladies des plantes sans les abîmer dans ce cas? Est-ce qu'on,
Alors, c'est vraiment de l'imagerie. Je peux vous tutoyer, Fella. C'est
bien
beaucoup plus
sûr, bien
pour
sûr,
moi.
bien
Je
sûr.
t'explique. Au fait, ces capteurs ou ce thermomètre, hein, pour reprendre un petit peu les mêmes termes que toi, donc c'est justement, c'est comme nous, la température, on a la température du corps quand on a la température qui dépasse justement 36 degrés ou 37, ou voilà, on est malade. Donc, c'est un peu pareil pour les plantes. Par contre, les plantes, parce que j'ai précisé la fluorescence, c'est-à-dire c'est la lumière qui est reflétée, au fait, les plantes, tous les corps, ils absorbent une lumière et on rejette une quantité. Donc, ce capteur-là, il va venir analyser cette réflectance. Et donc, c'est comme si c'est des images qu'il va avoir sur l'état physiologique de la plante au sein de la cellule et plus précisément au sein de où se fait la photosynthèse, hein, au niveau de la chlorophylle et donc et des pigments végétaux. Et donc, s'il y a des anomalies, il va le savoir. On peut le savoir. Il y a un tableau de bord. Donc, ce thermomètre-là, encore une fois, il va récupérer la donnée et on a un tableau de bord, on peut lire nos données. Donc, les pigmentations sont des indicateurs pour l'état physiologique de la plante. Et donc, c'est comme ça qu'on sait que quand il y a une anomalie dans la photosynthèse, donc ça veut dire que la plante, elle souffre. Déjà, on peut savoir sur le stress de la plante. Après, est-ce qu'il est biotique ou bien abiotique? Tu vas me dire, est-ce que c'est physiologique ou alors c'est pathogène? Là, dans ce cas de figure, il y a l'IA qui arrive, c'est-à-dire c'est un entraînement, un apprentissage automatique, l'IA, pour faire des modèles prédictifs selon les maladies. Alors, les maladies, nous, quand on voit la maladie, par exemple, moi, j'ai été formée, je travaille sur les alternarioses. Alors, l'alternaria, je connais très bien la tâche nécrotique de l'alternariose. Comme le mildiou, le botrytis, etc., toutes les autres maladies. Donc, on les reconnaît à travers les tâches, à travers les symptômes visibles. Par contre, le modèle IA, il va être fait selon les dommages physiologiques au sein des cellules. Donc, c'est comme, on va dire, l'IA, donc c'est un modèle qui va reconnaître l'alternaria, un autre modèle qui va reconnaître le mildiou, le botrytis, etc., etc. Donc, c'est un peu ça qui fait que on est en train justement de développer, ça prend un peu de temps, mais je suis dessus. Et donc, produire un peu des modèles qui vont reconnaître est-ce que c'est de l'alternariose, est-ce que c'est du mildiou, est-ce que c'est du botrytis, etc., etc. Donc, c'est selon les dégradations cellulaires et physiologiques de la plante. C'est comme ça que sera fait justement ce modèle-là et qui va nous dire, bien évidemment, c'est toujours nous, l'IA, et moi j'ai assisté hier à la première séance, j'ai oublié le professeur de Tlemcen qui a dit que l'IA, elle va pas nous, c'est nous qui commandons l'IA en fait, c'est nous qui ont créé cette technologie, et donc c'est nous qui restons patrons et c'est-à-dire meneurs de cette technologie. Donc nous, on est derrière justement, moi par exemple, je connais telle ou telle maladie et donc c'est moi qui vais aller justement derrière les prédictions des modèles de l'IA. Et c'est comme ça que je pourrais savoir produire des modèles qui demain, moi, écoute, je vais pas à me casser la tête. Est-ce que c'est de l'alternariat? Il y a une machine qui va travailler pour moi, qui va m'assister et qui, en plus, peut me dire à l'avance est-ce que je vais avoir telle ou telle maladie pour que moi, je puisse intervenir à l'avance et ne pas utiliser des produits chimiques. Bien sûr, je vais en utiliser parce que je dois traiter, comme si moi je suis malade, il faut que je prenne un paracétamol, mais je vais prendre juste une petite quantité, c'est-à-dire je vais traiter juste là où il faut parce que ce modèle-là m'a prévenu et me l'a dit bien avant que le champ ou ma parcelle soit totalement infestée.
Très bien. Je vous remercie, Dr Ayed. Vous avez répondu à ma deuxième question.
Merci à toi.
Ça veut dire détecter plutôt les maladies.
C'est l'objectif.
C'est le but.
Voilà. C'est le but. Au fait, ce sont des modèles qui vont nous prévenir bien avant l'apparition des symptômes. Dès qu'il y a des dégradations physiologiques au sein des cellules des végétaux, parce que nous, on ne les voit pas. Au fait, c'est une imagerie, c'est l'imagerie, il va scanner la plante Et il va voir au-delà du visible. Parce que nous, qu'est-ce qu'on voit? On voit au-delà du RGB. Nous, on voit le rouge, le bleu et le vert. Il y a des machines qui vont voir au-delà de ça. Donc, elles vont voir au sein de la cellule ce qui se passe physiologiquement. Donc, lorsqu'il y a détection de détérioration, on a une alerte. Et on a une alerte sur notre tableau de bord selon les pigments qui se dégradent. Est-ce que c'est la chlorophylle A? Est-ce que c'est la B? Est-ce que c'est les polyphénols? Est-ce que? Et nous, selon ce qui se dégrade au niveau de physiologique de la plante, on sait est-ce que la plante, elle est en train de produire des phytoalexines, c'est-à-dire elle se défend face à un pathogène, ou alors c'est une carence, c'est physiologique. En fait, c'est tout un domaine. Et nous, on est prévenus. Et donc, on va intervenir à temps et on peut apporter une solution à cette anomalie avant que le champ ou la parcelle soit envahie, quoi.
Très bien, très bien. Merci bien.
Avant l'apparition des symptômes.
Oui,
Madame Hidaïkra.
Je vous remercie.
Merci.
Madame Hidaïkra, est-ce que
à
je
vous.
peux poser
Oui.
une question?
Oui, bien sûr, vous pouvez.
Oui,
On a le
merci.
temps.
Donc, je remercie Madame Ayed, Docteur Ayed, pour cette
Merci
présentation
beaucoup.
très intéressante.
Shukran.
Donc,
Merci.
si j'ai bien compris, vous proposez un modèle. D'abord, j'ai deux petites questions. La première, ce modèle donc va remplacer le travail des phytopathologistes, en quelque sorte. D'accord? Et la deuxième, est-ce que c'est un modèle, ce que vous proposez, est-ce il a été testé sur terrain? Et comment vous voyez donc l'application à grande échelle de ce que vous proposez? Parce que c'est très intéressant, vu que nous avons souvent des maladies sur terrain, surtout quand il y a une culture intensive. Est-ce que c'est un modèle qui va s'appliquer à grande échelle?
Oui, madame, bien évidemment, c'est le but. C'est le but.
Voilà.
Au fait, vous savez, la technologie, elle avance tellement vite que c'est-à-dire aujourd'hui, avec l'ère du numérique, maintenant tout le monde est connecté. On ne peut pas vivre sans connexion, on ne peut pas vivre sans technologie, dans tous les domaines, et pourquoi pas le nôtre. C'est-à-dire, nous, on est en train d'explorer, de voir jusqu'à où va nous emmener cette technologie, jusqu'au niveau où elle va nous donner un grand coup de main dans la prédiction des maladies. Donc, elle ne va pas remplacer, pour répondre à votre première partie de questions, les phytopathologues. Donc, le phytopathologue reste, c'est lui qui va faire des analyses plus poussées, notamment moléculaires, quand c'est nécessaire. Donc, ça ne veut pas dire que les tests moléculaires vont disparaître, bien au contraire. Hein. Donc, travailler sur l'ADN, c'est toujours le dernier maillon pour, on va dire, qui est qui. Hein. Mais cette technologie numérique va précéder cette étape. Elle va justement donner un grand coup de main pour le phytopathologue pour lui dire attention, il y a une alerte du mildiou, par exemple. Et donc, cette technologie, elle va voir la venue de la maladie avant que le phytopathologue la voie. Parce que le phytopathologue… Pardon?
Je vous ai dit avant que le phytopathologue ne fasse le reste. Et c'est des tests ou des examens à la limite préventifs, voilà, qui nous permettent de gagner du temps.
Exactement, madame. Et c'est applicable à grande échelle parce que, comme l'a évoqué Walid tout à l'heure, notamment pour les drones et les systèmes embarqués, eh bien, les drones, ils vont embarquer des capteurs, ils vont survoler un champ de blé, par exemple, et ces capteurs-là vont prendre des acquisitions d'images sur le champ de blé. Donc, ils vont récupérer de la donnée. Cette donnée, elle est transférée sur un logiciel qui va justement analyser cette donnée et on peut savoir, par exemple, s'il y a de la septoriose ou pas au niveau de ce champ de blé. Donc, effectivement, c'est ça le but. Et pas que, parce que le drone est équipé de ces capteurs-là. Quand il va survoler un champ de blé, il va donc nous récupérer donc des acquisitions d'images qui seront analysées et on sait exactement où se trouve l'attaque. Par exemple, dans telle région, il y a un départ de septoriose. Donc, nous, on va aller exactement là où va partir la septoriose. Donc, on va traiter à ce niveau-là.
Parfait.
Et pas que. Il y a aussi des capteurs qui vont suivre les températures, l'humidité, parce qu'on sait très bien que les maladies, donc, elles adorent certaines températures et certaines humidités, hein. Et donc, elles sont fragiles à certaines températures et à certaines humidités. Donc, quand, par exemple, il y a un climat favorable pour le développement de telle ou telle maladie, on est alerté. Et donc, on peut intervenir bien à l'avance. Et c'est ça qui fera que, effectivement, on peut soigner, si je peux utiliser ce mot, on peut traiter notre champ et éviter le pire. Sans abuser ou sans utiliser beaucoup de produits chimiques. C'est-à-dire, moi, traditionnellement, qu'est-ce qui se fait quand on a une maladie? Eh bien, on va pulvériser tout le sang.
Oui.
On va
C'est ce qui
utiliser
se
Exact. Le but est non. Non, justement. Pour réduire l'utilisation de chimiques, il faut que j'aille utiliser uniquement là où se trouve le problème.
Très bien. Donc, ma dernière question aussi, avez-vous
Oui, madame.
pensé à l'impact économique? Parce que ce modèle-là, ça va nous faire gagner énormément c'est-à-dire le côté rendement, le côté économique en fait. Tout ça va tourner autour de l'impact économique et ça va
Eh bien,
être
oui,
très
madame.
intéressant.
Eh bien, oui, madame, c'est dans le contexte de l'agriculture durable.
Voilà.
Bien évidemment. Et en ce mot-là, uniquement l'agriculture durable, qui veut dire effectivement qu'il y a moins d'impact environnemental. L'agriculteur est gagnant, donc il va assurer son rendement sans impacter et l'économie et l'environnement. C'est totalement l'objectif, au fait. L'objectif est que l'agriculteur aujourd'hui, il peut pas faire. Au fait, on peut aussi parler de l'agriculture bio et de. Franchement, on a besoin de technologie, hein. Pour avancer maintenant, on s'est rendu compte qu'il faut arrêter l'utilisation de chimiques, il faut limiter l'agriculture intensive. Et pour aller dans l'agriculture qui est plutôt bénéfique à la fois pour l'agriculteur et le consommateur, pour la santé humaine, il faut utiliser cette technologie qui va nous assister dans cette démarche-là, tout simplement.
Eh bien, parfait. Je suis très convaincue. Donc, je vous souhaite également une bonne continuation pour votre travail qui est très intéressant. Bravo.
Merci beaucoup, Madame Chérif. Merci.
Au revoir.
Au revoir.
Donc, s'il n'y a pas d'autres questions, nous remercions vivement Docteur Tita. Ah, je te remercie.
Merci beaucoup, Madame Atika. Franchement, je vous remercie, mais vraiment beaucoup de m'avoir contacté, de m'avoir permis non seulement de participer et de partager un petit bout de ce que je fais, mais surtout de retrouver ma communauté et voilà mes confrères. Et voilà, c'est franchement, j'ai vraiment chaud au cœur. Merci beaucoup.
Le plaisir est pour nous.
Merci, Madame.
On passe à la prochaine conférence. We are going to move on the next conference of Professor Fethi Rabah. Are you here? Professeur Rabah, are you here? Vous pouvez lever la main, s'il vous plaît. Professeur Belrabah, je l'ai vu tout à l'heure, je l'ai invité à passer dans la session pénéliste. Vous pouvez lever la main pour que je puisse vous repérer, s'il vous plaît. I don't found it. Donc, on appelle la prochaine conférence. Donc, c'est docteur Rafef Nedjari de Tunis. Êtes-vous là en attendant professeur Belrabah? Peut-être c'est un problème technique dû à la connexion Internet. Donc, nous avons deux conférences qui se sont décommandées. Alors, docteur Morad Ounissi, qui s'est excusé, professeur Souad Mili Talbi, qui est actuellement en Allemagne, un stage. Donc, elle n'arrive pas à se connecter. Et donc, j'ai reçu plusieurs messages. Elle a essayé plusieurs fois de rejoindre le webinar, mais c'est impossible pour elle, ni par téléphone, ni par son PC. Non, je ne comprends pas pourquoi. Docteur Nesrine Sadawi aussi, elle s'excuse suite à des contraintes donc personnelles. Sinon, ceux qui n'ont pas pu se connecter, je vais voir que je travaille en même temps, hein. Donc, euh, puisque ils n'ont pas pu joindre le...
Même le docteur Moradoni ici, il a
Oui,
pas
il s'est
pu.
excusé hier. Il s'est excusé hier.
Ah,
Donc,
c'était
il y a
très
deux
compliqué.
communicants. Il y a professeur Mili qui n'arrive
Très
pas à se
difficile.
joindre. Elle est en Allemagne. Donc, elle n'a pas pu du tout accéder ni à aucun des liens que nous avons envoyés. Je ne sais pas ce qui se passe. Euh, c'est pas grave. بمكتب شغلها ça. Euh, je vous dis, puisque même Nedjari, donc aussi, elle ne m'a pas répondu par rapport à sa participation
Présentation.
avec une conférence. Peut-être il y a certains conférenciers qui ont demandé un autre jour parce
C'est
qu'ils
pas
ne peuvent pas, ils n'arrivent pas à se libérer, sachant que,
demain. Rataya, demain, c'est déjà.
oui, à
Demain, c'est
l'étranger,
déjà.
hein, comme Dr Afef Nedjari, elle est tunisienne, c'est notre collègue tunisienne. J'espère que c'est rien de grave. Nous les invitons.
Passer aux conférences,
Ils sont
aux
programmés.
conférences de l'après-midi.
Non, ils ont le
Pour,
lien de
par exemple, le
l'après-midi.
docteur Meliam Taleb. Là, non, c'est dommage.
Euh, ils, s- ils ne peuvent pas nous joindre. Le problème, c'est qu'ils n'arrivent pas à nous joindre.
Même le Xaibia, docteur Xaibia, docteur Taleb.
Non, ils ne peuvent pas puisqu'ils sont
Ok.
programmés. Ils sont programmés cet après-midi. Donc,
Ok.
ah, il y a un spectateur. Attendez. Ah, Naït Béchir. Vous pouvez parler, docteur Naït Béchir. Alors, malheureusement, tu arrives à la fin. Donc, nous allons donc passer à, donc plutôt nous arrêter là. C'est la fin du webinaire 3, hein. Et on se retrouvera, Inch'Allah, au webinaire 4 cet après-midi, Inch'Allah. Donc, est-ce que tu as quelque chose à dire, Monsieur Nate Bachir? Si tu peux nous présenter une conférence, donc tu es le bienvenu, hein, marhaba. Ah, je t'entends pas, je t'entends pas.
Bon.
Le micro, tu as éteint ton... Voilà, maintenant on
Ah,
peut
pardon,
t'entendre.
excusez-moi. Donc, excusez-moi d'arriver en retard. Euh, on avait programmé aujourd'hui, j'avais tout un programme ici, donc dans mon lieu de stage. Alors, déjà, merci beaucoup, Madame Daïkra, pour l'organisation. Merci beaucoup pour tous les efforts. Merci à toute l'équipe qui est en train de travailler ben, sur le, sur l'organisation de l'événement. Et sincèrement, je ne sais même pas quelle heure il est en Algérie parce que je viens de finir. Je suis complètement déboussolé.
Il
Mais
est
après,
11h45.
d'accord. Donc, il y aura une
11h45.
session pour l'après-midi, Inch'Allah.
Inch'Allah, oui.
Donc,
À
voilà. Donc, pour
14h.
l'après-midi, Inch'Allah. Très bien. Pour l'après-midi, donc, je serai présent, Inch'Allah.
Inch'Allah, Inch'Allah.
D'accord.
Donc, on vous remercie pour
Merci beaucoup. Merci à tous et
cette
à tes confrères.
intervention. Et Selmek. Alors, je vous dis à cet après-midi, Inch'Allah, et donc on se retrouvera. Donc, le webinaire 3 a pris fin et on passera, Inch'Allah, au webinaire 4. J'espère que euh, tout le monde peut se connecter facilement, Inch'Allah. Voilà. Alors, au revoir et à cet après-midi, Inch'Allah.
Merci beaucoup. Merci à